Okay, this is the microlife of Lake Attitash. So this photo my friend actually took on his drone. I thought it was really cool with all the boats on the lake and everything. So a little bit about Lake Attitash. It is a man-made lake. It was originally a pond that they placed a dam in and became a lake. This was around like the 1800s. It is a 1 square mile lake with a maximum depth of 34 feet, and it's average around 30 or 10 feet deep. It is considered a eutrophic lake due to farming too close to the watersheds, and this increased the levels of phosphorus, which resulted in an increase in cyanobacteria blooms. So twice a year, we have an alum treatment put into place in order to suppress the phosphorus levels and to prevent cyanobacteria from blooming. So I found this paper on how bacillus plays a role in soil, which can then help in understanding how the bacillus plays a role in lake sediment as well. Um, there are certain species of bacillus that can convert indigestible phosphorus into digestible phosphorus for plants. So relating that back to Lake Attitash, the bacillus are able to digest the high levels of undigestible phosphorus and convert it into digestible phosphorus, so the plants are able to use it. And there are also found species of bacteria that aid in nitrogen fixation, which can also help with cyanobacteria blooms. So I'm interested to see if these certain bacteria will show up within the taxonomic classifications due to these both playing a role in cyanobacteria blooms. If you are interested in finding out like how the bacillus plays a role in this, I do have the paper linked at the end of the slides. So the second paper I found was testing antibiotic resistance on bacillus. So they used multiple antibiotics against 6 strains of bacillus. So during the experiment you'll find later, I actually subcultured, I believe it's either Bacillus mycoides or Bacillus pseudomycoides. So I want to test the antibiotic resistance towards my subculture and see how it reflects this graph that when they did the experiment. So I will refer back to this later in the presentation. So obtaining lake water, I collected 6 samples of lake sediment. I did the West Shore Middle, which is in front of my house. And then I did West Shore Left, which is just closer to the runoff, so like where the Powwow River is. And then I did West Shore Right, which is kind of closer to where the other watershed is. And then I did the only public beach on our lake, which is over here. And I did the dam, which is right here, which is the closest to the watershed. And then I did the boat ramp, which is right here. So I collected the sediment, and I did collect all of these, but I only used the sediment for West Shore Middle because this is in front of my house. I spend the most of the time there. So culturing the sample, I used a clipped micropipette and placed 200 microliters of sediment and placed onto a triptych soy agar. I used the hockey puck shaped tool to spread the sample around evenly, and I did 5 plates and then placed into an incubator at 30 degrees Celsius and checked after 24 hours. I tried to do a generic approach because I was not aware of what was in my lake. I wanted to see what I could grow. So this is the most generic approach. After 24 hours, I did take an inoculated loop and scoop some cells to be placed into the tubes to be DNA tested. So this is an overall view of the microbes found within the sample. So majority is Pseudomonas, which is predictable because this is like a public water. And there is a good amount of bacillus. So at first I was going to look at what type of Pseudomonas was in the lake, but I wanted to gear more towards the bacillus because that is what I think I subcultured. So this is the Terrabacteria group. I'm only including this because I wanted to show that I did grow cyanobacteria, which is really interesting because we do the alum treatment so often. I wasn't sure if we were going to find cyanobacteria in there or not. So it is interesting that I did find a little bit. So this is a chart of just the overall chart of bacillus present within the sample. There's a lot. So I tried to narrow it down a little bit to just the significant types of bacillus there. So that is Bacillus cereus, Bacillus luti, Bacillus thuringiensis, Bacillus myers flavi, and Bacillus pseudomycoides. So I wanted to take those and compare it back to the bacillus that they were talking about in the soil. So Bacillus cereus and Bacillus circulans both aid in phosphorus digestion and nitrogen fixation, while Bacillus mycoides and Bacillus subtilis just aid in phosphorus digestion. It was really interesting that I only found this many Bacillus species in my lake. I really thought I was going to find more because the phosphorus levels are such a big issue. But it is interesting. I did find a little bit. I was kind of surprised I didn't find any that aided in just nitrogen fixation too. But moving on. So I then subcultured. So referring back to the culture slide, which I can bring up. I chose to subculture this fuzzy looking colony just because I thought it was most interesting and it stood out the most to me. And I thought it'd probably be the easiest to subculture too. So I used the inoculating loop and scooped up the colony and streak plated onto tryptic soy agar and then placed it into the incubator at 30 degrees Celsius. I came to a pure culture pretty quick, only after streak plating around 3 times. And I think that's just because it's you can really tell which is the bacillus species. And it's found to be a filamentous colony. Species 1, subculture 1 seems to have 2 microbial species. So you can see the filamentous colony, but then you can see non-filamentous colonies right here, which means that there's 2 species in there. But this subculture 3, it just seems to be in pure culture. Even that colony over there seems a little filamentous to me. So then I went on to gram staining. So I started with a gram stain control, and I gram stained Bacillus megaterium and E. coli. Bacillus megaterium is known to be a gram-positive bacteria that stains purple, while E. coli is known to be a gram-negative bacteria that will stain pink. So this is a photo that was taken around 1,000 magnification. And so you can tell right here, the purple is the Bacillus megaterium, and then these small little ones are E. coli. So I based this off of when I gram stained mine to see if it's a gram positive or gram negative bacteria. So I use the cells from the subculture to gram stain, appear to be purple, which means that the cells are gram positive. So the photo on the left was taken at 1,000 magnification, while the photo on the right was taken at 400 magnification. So yeah, they're definitely purple. They're definitely gram positive. So then I went on to do a phase contrast microscopy, and I did not have an issue at all finding the cells underneath the microscope because they are fairly big compared to other bacteria when using the phase contrast. The cells appear to be rod shaped, and the size appeared to be around 0.25 to 0.3 micrometers. And yeah. So then I went on to doing the antibiotic resistance to the subculture. So I placed the subcultured cells into 2 tubes of broth and placed the tubes in the shaker for 24 hours at 30 degrees Celsius. I took out 4 tryptic soy agar plates and divided them into 4 quadrants for each of the antibiotic names with the control, and in total 8 were used. So you can tell on these 2 plates that I divided them into 4 so I could add all of the antibiotic names. I placed 150 milliliters of the broth with the cells in them now onto the tryptic soy agar plates and added each of the antibiotics to the corresponding quadrants. So I did the same thing that I did when I streaked my lake sediment. I used the hockey puck tool and I spread it around evenly, making sure that the cells did get into the agar and allow them to grow. After I did put the corresponding antibiotics to the correct plates, I placed it into the 30 degrees Celsius incubator because that's what I've just been using this whole time. And the 2 plates that show results of the testing, I believe that the other 2 plates are better results. I don't think they're just got that many cells onto these two. So this is a better one. I think you can see more of how the bacteria interacts with the antibiotics. So comparing the results with the bacillus antibiotic resistance, on the plates, the antibiotics that produced the most prominent zones of clearance, so they had the least resistance to the antibiotics, was tetracycline, neomycin, erythromycin, and chloramphenicol. And the kanamycin and the penicillin seem to have no resistance to the bacillus strain, along with the streptomycin. It's inconclusive due to the lack of microbe growth on both of the plates. So this one only has like a couple of colonies forming around it. Well, same with the other plate as well. It doesn't seem like it is a little bit resistant, but you can't conclude it. Oops. So focusing on the graph, so I can bring that back. The tetracycline, erythromycin, and the chloramphenicol had no resistance with the Bacillus mycoides and the Pseudomycoides, and both penicillin showed full resistance to it. It does seem that the culture or the subculture I have leans a little bit more towards the Bacillus pseudomycoides, also because that was more of the Bacillus pseudomycoides did show up more into the taxonomic classification. If you see right here, there's like 2%. Oh no, 6%. So I'm not sure which one. Both of them look very similar on plates. So you should probably have to do a DNA extract and just really determine which one is there. So in conclusion, the point of the experiment, I really just wanted to see what microbes were found in Lake Attitash and grown under certain conditions. The Pseudomonas was the most common microbe found in the sample. However, I did subculture Bacillus, and the gram stain and phase contrast showed how the cells appeared, and it did correspond with the Bacillus strain rather than the Pseudomonas. The antibiotic resistance test also helped in discovering what Bacillus was subcultured. Not sure if it's mycoides or pseudomycoides. So they are very similar to one another. And yeah, I did really like this experiment. It was really interesting. But I mean, you really can't conclude these were the only microbes living in the lake. I subcultured it in February when it's really cold, so you don't know if there's different microbes that grow when it's really cold. Like there was still ice in the lake, and like it'd be interesting to do again in July and see what difference it is. It would also be interesting to incubate it in like a 37-degree incubator and see if there's difference in microbial growth, because obviously there are some bacteria that grow under certain conditions. But yeah, I'd like to thank the Dr. Launen for allowing us to do this experiment and have such free range with it. I'd like to thank UNH for funding it and doing the taxonomic classification. I'd also like to thank Keene State for providing all of the tools necessary to perform this research. So yeah, thank you for watching. Here are both my references if you are interested in seeing or reading about the bacillus that interacts with the soil, and the other one is the bacillus that they did antibiotic testing on. So yeah, I didn't go too much into it, but these two will definitely provide a lot of information for you. So yeah, thank you for watching.